sea turtles, whales, and even this guy, the boggenmoth.
With bufo and rana temporaria, we didn't have the spectrosensitivity.
Hello, my name is Marcel Savre. So I'm a PhD student working with Stanley Heinz at Lund University. And right now I'm at the Visionarium at Tvargmina Zoological Research Station. The Visionarium is a conference by which people who study a wide range of things, mostly vision, can come and present their research in the format of a poster, as I have done here, or as a lecture. And it's a great, wonderful conference, and I highly recommend anybody visit it if they're able to. So I talk about my research right now really quick. So we study insect brains. And so basically we study brains because we want to understand how the brain is able to drive behavior. And so we use insects as kind of a model species to be able to ask these kinds of questions. So insects, being one of the most species-rich phyla on the entire planet, they occupy a large range of different kinds of habitats. And so they have evolved different mechanisms for being able to navigate in these kinds of habitats. And so, for example, a solitary nocturnal bee, for example, the sweat bee, the megalopta, these bees live in dense forests, and they have a nest that they return to, so they go out and forage, and they don't have very much information like from the sky or from the sun or from the moon because they're nocturnal. So they use a lot of visual local cues, whereas something, for example, like the desert ants, they live in vast plains, vast desert plains, and all they have to be able to navigate is the location of the sun in the sky and the polarization pattern that's emitted from the sun, and possibly panoramic view of the mountain ranges, if those exist, around. And so you can see that these insects, they live in these different kinds of habitats, and they use different kinds of visual cues to be able to navigate within them. There's differences also in terms of the locomotion, so ants typically will walk when they're foraging, whereas bees fly from various places. So the question is how are these different navigational strategies represented in the brain of insects which use different navigational strategies? So basically what we do is we take the brains of insects, and we primarily focus on bumblebees and ants. And so we use a technique called serial block-based electron microscopy. And basically what we do is we image the brain, and we create a stack of a bunch of two-dimensional images that become a three-dimensional image, which we can then trace and use to identify individual neurons and trace them. And the goal is to do a comprehensive tracing of every single neuron that is involved in the brain region that's involved in navigation. So in insects, this is a brain region that's known as the central complex. And the central complex is a highly conserved region that exists in all insects. And it's known to be involved with things like action selection and sensory integration, and particularly navigation. So the idea is, once we have this neuron anatomical map, we can then begin to model how we think this circuitry works. And so we can create computational models, and we can actually even employ these computational models into robotic interfaces, and we can create, for example, a small car that can actually do a task called path integration to be able to simulate foraging in an inside environment. So then once we have these models, we then test and verify these models using a method called electrophysiology, where we basically take an electrode and we stick it into the insect brains, and the insects are inside of an arena, and we provide them with some kind of stimuli. And we record from individual neurons while we provide them with the stimuli to see how they react. So then we can basically have data about how this circuitry works, and we can confirm it and compare it to the model that we have created. And so in the end, what we hope to do is to be able to understand better how simple elementary decisions can lead to this complex and elaborate behavior that we understand as foraging, and ultimately hope to understand how the brain is able to drive behavior in a more general sense.